Good afternoon. My name is Kiyoizumi, Executive Vice President of Thrive Bioscience, located in Boston, and I have an office in San Diego as well. The Thrive started the business about 4 years ago with a special mission, so to speak, to automate the cell culture. This is a very different concept. I think cell culture is a part of R&D project. Pretty much everybody does it every day. But we aim to automate and make it a consistent and controlled procedure. And it's especially very important for stem cell culture as well. No, this one. Let me start with the highlighting the key points about Thrive. Cell culture is at the center of biomedical research and cell therapeutics, as you know. This is a very large market, almost $19 billion annually spent in cell culture. And it's conducted over 200,000 non-smart incubators in 80,000 labs across the world. And today, cell culture is done basically the same way as it was done 65 years ago. It's basically a manual procedure. And the significant pain points still exist and actually increasing more based on the more cell lines available based on CRISPR technology. Thrive solves these pain points with instrument. We will start sales later this year and with no FDA approval required. We project our revenues growing to over $100 million annually in 2023 with a significant portion with recurring consumables and software. We have two significant collaborations in this area with the Broad Institute of MIT and Harvard and the Harvard Stem Cell Institute. Broad, the researchers found that even short period of time that the cell culture, doing the cell culture causes genomic drift from stress. This makes it very difficult to conducting the quality drug discovery research consistently. And the Harvard Stem Cell Institute is automating the difficult tasks in growing, especially the iPS cells. We have built a significant IP portfolio with 42 patent applications, of which 7 are already issued. And because no one has built these intelligent cell culture instruments, we have very limited competition in this neglected field. We have raised $18 million to date, and our investors are from 7 angel investors groups, including life science angels, executives from such as former CEOs of Novartis, Cytek, and Exact Sciences, and family offices such as Estee Lauder family. So we have been raising pretty much angel group or individuals to date. And we are starting investors. We're looking for investors for Series B round. We plan to close by the end of this year. And if you see the picture on the bottom, 1952 is the cell line factory at Takashi Institute and growing HeLa cells. And I'm sad to say the procedure or process hasn't been much improved since that time. So Thrive's products are providing analytics, automation, and hence better technology. So what the cell culture? I think probably most of you are familiar with cell culture. So I just make it brief and the point of the pain points. So cell culture is trying to recreate the cellular environment outside of the body, basically in vitro. That's the try to create as physiological as possible. The cells are currently growing in so-called, we call it non-smart incubators. It's a box with controlled temperature and the gases, basically CO2 and oxygen. But they don't control another important factors such as pH or nutrients. As you see, we are adding the capability of intelligence into these incubators. That's our mission. And in order to even look at the cells to run experiments on them, they are moved across many environments, basically taking out and the temperature, changing the culture medium, then put it back. So that change of the controlled environment into uncontrolled environment put a lot of stress on the cells. And another issue we see is when the researchers take out the cells, look at the microscope and counting the cells, etc., then put it back. Of course, they record how many cells were there and we change the medium, but they don't record any images. Image capture is not there. So depending on the researcher, the subjective observation is all over the place, and it's not controlled. So we try to standardize and control environment for the cell culture. And another important aspect is the cells are very delicate, and they drift genomically under different stress or circumstances, environments. And the cells are also mislabeled, tend to be mislabeled and prone to contamination. And a recent NIH study stated 15 to up to 36% of the cells are mislabeled. And this means that the researchers think they're dealing with specific type of cells, but in fact, it may not. So that's a lot of waste and very, very inconclusive study results we've seen. And it's not reproducible. So because cells can drift genomically and stressed, they behave differently across within the labs, and all the different results, as you know, when you see the published papers, reproducibility may not be there necessarily, and it's very controversial. You cannot replicate the result of certain research. So good science needs good cells. That's our belief, and we try to provide good cells to any research environment. So our solution is right now we are building 2 instruments. And the first one is cell assist on the left. And this is, we call it the cell metrics in a box. So it's currently in beta testing at Harvard and Broad Institute at Harvard and MIT. It's a benchtop solution that improves existing cell culture incubators. So you use with current conventional incubators, but you add cell assist in terms of intellectual part, the brain part. And this we are selling around $50,000. And we are starting early stage adoption sales towards the end of the year. And the second product is Alpaca. This is fully automated, basically cell culture in a box. So the cells never leave this Alpaca instrumentation. And this reduces any stress, changes of environments, and Alpaca is priced around $500,000. The current practice of cell culture is that the researcher takes the cells out of the box and put all the procedures and put it back. And instead, with cell assist, the researcher puts the culture plates into cell assist, and the cell assist will analyze the image, capture the image, and provide the researcher with the feedback of instructions to manage cell culture more quality. And we are developing the first time in biology major database for the labs to capture images and how to interpret the image and consistently. So that's another aspect of not only capturing the image, but also the interpretation analysis we provide. So that the researcher can go back, if something wrong, they can go back to see the image and why it didn't go as before, or what was the factors, change of the medium, lot size, whatnot. So we can track those. Let me talk about commercial opportunity for Thrive. As I mentioned, the cell culture market is quite large. Because it's kind of quiet market. Everybody uses every day, so that they don't really think about it. It's mostly consumables and some of the hardware and software as well. And about 3/4 are industry use, and 1/4 is academic researchers. And the application can include very wide varieties of activities, creating cell libraries, assay development, in vitro toxicology, of course, and recently, especially the iPS cells or stem cell culture. That's creating a very important component of cell culture. Let me touch upon the management of Thrive Bioscience. CEO Tom Farhorsh is a serial entrepreneur, and he has started about 10 companies in the past, including Exact Sciences, Cytic, and HNC Software. Those have a successful exit today. And we have many of the same investors, fortunately, with Thrive, and the employees also as a part of the management team. Alan Blanchard is the CSO and the scientific founder, and he's responsible for some of the many of the important inventions, especially in sequencing the area, including Illumina's technology and many of the synthetic biology companies' technologies. And his vision of Thrive is such that after spending 25 years in genomics, he wanted to bring the same amount of reproducible data to the cell culture world. And our board member also has a direct and relevant experience, including Guy Broadbent, our chairman, is a former president of, I would say, the largest laboratory product group in the world, the Thermo Fisher. Mike Finney, who was a co-founder of MJ Research and currently very active in angel investment with Life Science Angels, and Brock Reeve, the executive director of Harvard Stem Cell Institute. So let me summarize Thrive's opportunity here. Product development on schedule. We are a small company with 15 people, but we have done product development and currently beta site testing ongoing at Harvard Stem Cell Institute, Broad Institute of MIT and Harvard. And we are starting to add more beta sites at Stanford, Mass General, and other leading academic institutions. And the second, the large market potential. As I mentioned, cell culture is a large market, and we are going into update the cell culture procedure with our intelligent instrumentation. Excellent market traction, strong IP portfolio we have created, and a capable management team. And we have a track record of financing. We have successfully raised the money to date. So we are looking for investors for upcoming Series B round, and also would like to recruit beta site testing, both academia or industry, collaborate with us. And I welcome speaking with you after my talk to pursue the potential collaboration. And so we would like to, again, bring the automation, analysis, and better technology to the world of cell culture. Thank you very much for your attention.